Welcome to the MVP, the ModernVivo podcast. I'm your host, Andrés Velarde, Director of Operations at ModernVivo. And in each episode, I sit down with a different life scientist to talk about their research findings, the implications of those findings, as well as their career trajectories and the steps they took to arrive where they are now. Today I have a conversation with Neha Binish and Jonas Terlau, two researchers that recently published in Nature Neuroscience on their work regarding how different brain regions, namely the prefrontal cortex and the motor cortex, communicate with each other. Their work was conducted at the Hardy Institute for Clinical Brain Research at the University Medical Center in Tübingen, Germany. Stay tuned to hear more about their story and their findings. Hello, Jonas, Neha. Welcome to the show. I'm very excited for you to be here. Before we go any further into the research, do you want to just give brief introductions on your backgrounds?
Sure, Neha, go ahead.
Hi, I'm Neha, and I'm a PhD student here in Germany at the University of Tübingen, and I work with a group of Randolph Helfrich, and he is currently at Yale, and we work together on all things neuroscience in a way, if you think about it. That's it, pretty much. You can go ahead, Jonas.
No, it sounds pretty safe for me. Like we worked together in the same lab for the last 3 years, Randolph Helfrich's lab in Tübingen. I then moved to Munich a couple of months ago. And yeah, the main focus of our work is really like intracranial human recordings and cognitive neuroscience. And this is what this paper is also about, right?
Yeah.
Right.
Yeah, thank you. For our listeners, Neha and Jonas published a paper in Nature Neuroscience in May of 2025 titled "A Communication Subspace Relates Context-Dependent Actions from Human Prefrontal to Motor Cortex," which is a mouthful for people who maybe aren't super familiar with the brain. Can you say a little bit more about where this work originated and what brought you to it?
Right. So I come from a very technical background in a way. So when I came into Randolph's lab, we started talking about like, okay, how can I contribute in a way, right, to the questions that we were asking at the time. So one of the things that Randolph was very interested was the prefrontal cortex, right? So um, it's like part of the human brain that's very evolved in a way, and we think that it's somehow responsible for like influencing our actions and turning our intentions or goals into like behavior and so on. So it's sort of like what we thought of was like the control center of the brain, right? This is what's the narrative in the current literature of what it is, right? So the questions then that I had in particular was that, okay, so we look at the activity within like this region of the brain, and we find that it's really complex, right? So the sort of information that's encoded in the neural activity that we record from like intracranial patients in Tübingen, we see that it's very, very complex the way the information is encoded. And so if this region needs to control action, for example, how does it turn this really complex abstract information into something very simple, like a motor output, where you're probably doing something like pressing a button, for example, right?
Mhm.
So that's sort of how I started out thinking about my project and how this sort of evolved, right?
Yeah.
Yeah. And
I mean, yeah, if we want to take a step back and we can also first talk about why human cognitive neuroscience is really interested in the prefrontal cortex, right? So, and like Neha said, it's obviously it's a region like also other association cortices that has evolved and expanded massively throughout primate evolution. And actually, we are not really sure why that is. So there are many theories on it, but what we can say is that there has been like this gigantic leap in terms of complexity of behavior, right? That defines us humans compared to other animals. And I always like to think about this in terms of examples. So Andres, for you, it's now like about 8 a.m. So I hope you had some coffee already, right?
Yeah, I sure
Coffee
did.
making, right. So coffee making, I don't know how is it for you? Is it like a button press? Is it a simple machine, or does it involve some more
Oh,
complicated?
okay. It's mine is a bit more complicated because my coffee grinder recently broke. So you pour the beans whole into a mortar and pestle, and then I spend 5 to 7 minutes grinding the beans, and then I transfer them into a French press, fill that with hot water, wait 4 to 5 minutes, press, pour, and then I'm ready to go.
Exactly. So I think that's a fantastic example. So what you just described is actually a pretty complex sequence of actions, right?
Yeah.
And the interesting thing about this is that we know from behavioral studies that you can basically omit like single steps in the sequence and you're still able to perform the rest of it. So you won't end up having coffee, but you're still able to follow the sequence in a way that basically you choose the remaining steps of the sequence. You can perform them despite leaving out individual steps. So that means you're not just performing one step after another at a time. You're not chaining sequence steps, basically. But that means you have sort of a temporally extended abstract representation of this whole idea of making coffee. So the whole sequential action is basically an abstract representation in your brain. And what we also know is that patients who have lesions in the prefrontal cortex struggle a lot with this kind of performance or behavior. So as soon as behavior becomes more complex, involves more sequential ideas, and you have to really have this mental programming, okay, or where am I right now in this sequence? This becomes a lot more difficult once you have lesion PFC. And one other thing that we also know is like heavily PFC dependent is context dependent behavior. So when I talk about sequential action, the same idea, these temporally extended representations apply to your sensory environment. What that means is right now we're in this meeting, we're having a nice conversation. So if my phone starts ringing, I won't pick up. If I'm at home, phone starts ringing, of course I pick up. Hi, what's up, right? So that it's the exact same sensory event can evoke basically opposite responses based on the temporal context. And this basically is the same idea, right? So prefrontal cortex allows me to have this temporally extended representation of either the sensory environment or of any action plans that I want to perform. And this leads then to pretty complex and goal-directed behavior that is pretty unique to us humans. And that brings us actually back to the paper, because this is exactly what we want to probe with this task.
Yeah, I was going to ask. So, what was the context in which you kind of ran this experiment? Like, who were your subjects? What was the task? What was the kind of the goal-directed behavior you were looking at?
Right. So, Neha, maybe you can talk
Yeah.
more about this.
So with the task, actually, it's pretty simple, right? You just because we record from patients who have been admitted to the hospital who are like pharmacoresistant to epilepsy, right? So what that means is that at some point when people have epilepsy, they stop reacting to the drugs, so they don't have it starts having no effect on them, right? So then one of the next steps in terms of treatment is then you could kind of try to locate where the epilepsy starts from and try to either lesion that part out in the brain or cut out that part of the tissue of the brain, right? So these are the sort of patients that we work with, and they are, they're in the hospital for like 2 weeks or so with these electrodes that are implanted into their brain on like the cortical area of the brain. So on the cortical surface, where you can sort of like, record the electrical activity that's happening, right? And so the placement of these electrodes are like completely based on, for example, what kind of, it's like medically focused, right? We have no say in how it's placed and so on, right? But what we know is that, okay, there's this patient here at the hospital and they are, for example, they have like coverage within the PFC or like the motor area and so on. And so we go to the hospital, we have this set of like cognitive tasks that's prepared. For specifically our case, we had this task where it's very simple, where we have a laptop basically, right? And you flash a set of like triangles, right? And we tell the patients that if there's a specific sequence, then it will, like, if it's like, for example, left, up, and right or so, right? And then you tell them that immediately after the right, there will be a downward facing triangle, right? That's the context that we give them. And we tell them every time you see a downward facing triangle, you have to press a button, like the right key or whatever, right? The right arrow. And so it's pretty simple, right? So there are contexts where you can predict that a downward facing triangle is going to appear on the screen and then you can react to it. And there are times when it just randomly appears, right? So in the case where you can sort of prepare a response, we expect them to be faster. And where you cannot, they're kind of slower, right?
Yeah,
Based
makes
on
sense.
this context that you have, right? So that's the kind of task that we have. And it's sort of like a really niche recording that we have, right? The intracranial recordings is not very, you get like maybe one patient in a month or so. And this is sort of recordings that we work with. And I think it's really important because it's been shown that, it's on the cortical surface, unlike EEG or fMRI and so on, right? So you have a great temporal resolution for this kind of data, and you also can extract this high frequency activity from the electrical activity, right? And that's basically said to sort of correlate with some sort of underlying population activity, right? So these are the signals that we work with, and I think it's a really niche sort of recording. And
Yeah. When you
yeah,
say underlying population activity, you're talking about like groups of neurons in these cortices firing together. Is that right?
yeah, yeah. So
Yeah.
I mean, the origins of what it might represent is still very debated, right? We still don't know what high gamma exactly represents. For example, there is this new Nature piece that people can go and read where they talk about how it might be some sort of summed activity plus like this synaptic currents that is like integrated from like basically large regions where neurons fire together. But these are things that are still being debated in the field, I think, still. I mean, we use this as a marker because it's the best marker that we have. But at the end of the day, I don't know if we can answer exactly what it is.
Yeah, I mean, it's an ongoing journey. You'll have to send me this paper. I think that's interesting. Maybe I can include it in
Yeah,
the show notes for readers who are curious.
for
Um,
sure. Yeah.
yeah,
But
yeah. I want. Oh, go ahead.
yeah, I mean, I feel like I have to add that the sort of advancement in technology nowadays lets us record from different regions of the brain simultaneously, right? Before we only had like recordings from a single region of the brain, and you look at how that region is encoding information or whatever information that you want to know from the data, right? But now we have access to like simultaneous recordings from like multiple regions of the brain, and we're sort of like we can relate these sort of information, right? This is also what we thought about in our task, where we know the PFC is sort of like a control center, and we know the motor area is responsible for like producing action, right? How do these two regions communicate? So that's sort of like the basis of the whole paper, right?
Yeah,
Yeah.
certainly. And I wanted to kind of get into this, right? So there's this idea of a subspace, which to me is like quite hard to wrap my head around, right? It's this kind of theoretical information relay. It seems like you identified it using statistical means as well. Let's dig into that a little bit. Like, what is a communication subspace? How can we think about it in maybe more concrete terms?
Right. So, so like I just said, I mean, you have access to these simultaneous recordings now, right? So, which was not something that we had before. And what we can sort of look at the data and say is how do what sort of information is shared between PFC and M1, right? This is something that you can
Mm-hmm.
sort of quantify once you have data, right? And so it's sort of like looking at the PFC and then saying that, hey, what are the prominent patterns of activity within the PFC that seem to be predictive of what's happening in the M1, right? So you can
Mm-hmm.
look at the patterns of activity, right? Basically, now that if you combine all the channels that you have from the PFC and you look at like what's the most prominent pattern, and that is specifically important for what's happening in M1, you can pick out like a couple of patterns then, right? And then you see, okay, what are these patterns doing? So these patterns are basically what we call a subspace, right? And this is something that's like very well developed in non-human primates, the theory behind this, right? Where they show that you can pick out like prominent patterns between visual regions that are communicating and so on. So we wanted to sort of apply the same idea to humans and see if this is also the case for like regions like PFC, where we don't really know how it encodes information that's responsible to communicate with M1, right? Yeah. So that's sort of like the subspace idea. And then what we found is that we are able to recover patterns that kind of look very similar to how the M1 fires, right? So if you look at a channel in M1, you see a very prominent peak when you press the button, right? And what we see is that, okay, PFC, it doesn't show that same kind of activity, but once you look at the subspace of these prominent patterns, you see the same kind of motor pattern, which was really surprising to us in the beginning, I think, right?
Mm-hmm.
And then we were like, okay, maybe it's a mathematical artifact, right? I mean, you're kind of optimizing your model to sort of pick out this data, right? Is this even like a true effect, right? And so this is I mean, then we were like, okay, but how do we prove that it's maybe something that's actually important, right? And that's when we sort of like related this to behavior, and we see that, so in the case where you have the context, like I talked about, where you have the predictive sequence, where they're faster, you see
Mm-hmm.
that this pattern also sort of arises in the subspace a little earlier as compared to like the other ones where they're like a little later. So
Mm-hmm.
you're able to now sort of like temporally relate these prominent patterns within the PFC to your actual button press, that the behavior of the participant that was actually pressing the button, right? So yeah, this is, I mean, that's basically a whole paper. So we were like, okay,
Yeah,
oh,
so
that's
cool.
great. It's not an artifact, so we were kind of happy about it, right? Yeah, it's a
Yeah, thanks for sharing, Jonas. What was your take when you saw this data?
No, I just wanted like I also wanted to add something about the whole subspace idea because this has been
Mm-hmm.
like major idea and also a little buzzword like for the last couple of years now in neuroscience. Um, and like Neha said, it basically it began like also or it's heavily dependent on this situation that we're in right now to be able to record from hundreds of different electrodes, channels, or even neurons at the same time. And what you see then is that they do not do like the firing activity is not independent. So many different neurons tend to do correlated stuff. And what this means is then that you can find sort of linear mappings of this activity that basically capture this correlated activity. So you basically reduce the complexity of the data. And this
Mm-hmm.
is it's very important if you want then to do like different analyses on this. And then it's been shown, for example, that these low dimensional patterns remain extremely consistent across time, which is different, which makes them different from the firing patterns in the first place.
Mm-hmm.
And the main analytic approach to find these patterns has been principal component analysis. Principal components, so PCA basically finds or gives you these mappings that basically extract these low dimensional spaces where you can really also visualize the correlated activity of the population. And what we did here is related, but it's different in a meaningful way. So like Neha described, the way this subspace is designed is not that it maximizes the variance or the explained variance in the data, but that it really maximizes the way or the part of the activity in prefrontal cortex that is predictive of whatever activity is in M1 at the same time.
Mm-hmm.
And this is, of course, like conceptually motivated by this whole idea that we know that prefrontal cortex has to guide behavior through M1 in some way, right? Because this is like sort of your motor output. And of course, M1 is doing way more than just being this passive relay station, but this is the pathway that we want to understand here. And we know that this is also grounded in anatomy via subcortical pathways or via prefrontal premotor pathways, right? So, but there is very little evidence on how exactly this works. And there's even less evidence in settings like this where you really look at higher level cognition, like we did here.
Yeah.
So yeah, and this was like then, like Neha said, we looked at the data, we saw, okay, there are bumps of activity, there are peaks of activity in PFC and M1. So the next step is to show that this is really meaningful and explains behavior, and it's not just either a mathematical artifact or just the motor response in the first place.
Yeah.
right.
Yeah.
Yeah, interesting. I'm trying to think of a good analogy for it. Like, would it be accurate for me to consider like, okay, so what you've shown is that your prefrontal cortex is doing kind of like a information compression on its way to the motor cortex, and like only the information that is necessary and sufficient for the motor cortex to fire correctly is being sent? Is that what's kind of happening here, where it's like the prefrontal cortex isn't just dumping all this random noisy information to the motor cortex, but almost like you because kind of mentioned this like low dimensional pattern, right? A reduction in complexity. So am I thinking about that correctly? Is it like an information compression?
So the way I think about it, I think is more like, well, I cannot say if the PFC, like from the data that from the paper that we have, for example, I cannot say for sure if this activity pattern that we recover in the PFC, like you said, which is this compressed pattern or like this very, very prominent pattern, um, if that's actually driving the motor cortex, right? But what we can say for sure is that this compressed information exists within the PFC, and
Mm-hmm.
it seems to be linked to behavior, right? So this is, I think it's a little tricky to kind of claim that it's driving the motor cortex, because I mean we use the motor cortex to look at the PFC to kind of pick out these patterns, right? That's what we use the motor cortex for, right? And we obviously would like to believe that this is what's happening. But what we can say for sure now is that these patterns of activity exist and that it's correlated with the sort of behavior that we see. But correlation is as far as we can claim, I think, right?
Yeah.
Unless Jonas has something to
No,
say.
but I think the way that you described it is actually pretty accurate, right? Because like this is the starting point that we had. You look at PFC activity, you see this extremely high dimensional activity, and then the question is, okay, but what part of this is actually relevant for behavior? Because a lot of stuff is going on, a lot of like parallel processing is happening in the brain, and especially in prefrontal cortex. And what part of this is then actually relayed and communicated to downstream centers? And the communication subspace is sort of a functional way to define the subspace or pattern of activity that is actually relayed. And then the nice part is this is purely data driven. This is you just look at activity, you predict basically M1 based on PFC, and what you then find only in the next step is okay, but this mapping actually explains the behavioral effect without doing any labeled working beforehand.
Yeah. We also don't label the data by saying that, okay, so these are the traces that are faster or these are the traces that are slower, right? It's completely just based on like just sort of like correlating patterns between the PFC and the M1, right?
There's so much more, I think, with regards to just like this specific paper and experiment that we can dig into. But, I think I want to kind of switch gears, maybe zoom out a little bit more. And if people have questions about your specific publication, maybe they can reach out to you directly. But I wanted to talk about like the implications of this work. What's kind of the deeper so what behind what you found and, the research that will come after you as well? Like, what are your thoughts?
I think this is something that Jonas is very well suited to answer, considering he's working with BCI now, which I think
Cool.
is a very cool field.
Yeah. So right, this is definitely like a field where these ideas could become relevant in the future. So, like I said, like first the whole idea of subspaces is of course highly relevant because it's been shown, like I said before, that once you look at these low dimensional patterns of activity, you see that across time activity is much more stable, or at least you can find very simple ways to align activity then across different days, weeks, months. And this is highly relevant for chronic brain-computer interfaces where you want to have stable performance and you want to avoid like these very frequent recalibrations, of course, right? Because we know that brain activity is constantly shifting. This is like sort of called the representational drift. So, and dimensionality reduction of this activity or defining subspaces is a major topic in this field. And I think communication subspaces in general have only like emerged fairly recently, and they haven't found their way yet into any applications. But in my mind, it's really straightforward to not think about the brain in terms of isolated areas that do their like little computations and then generate behavior in some way, but it's an interconnected network, right? And we have actually fantastic imaging techniques to unravel these like the structural wide meta tracks and all there is, but in my mind, we lag a little behind on the like neurophysiological front because this has to become more of a multimodal approach where we integrate really the imaging stuff and what we know about the brain structure, and then also use techniques like communication subspaces where you can sort of more closely look at what's actually going on between these areas, because like interregional communication is definitely one of the major ways that the brain generates behavior and not just modular isolated stuff. That's happening.
I got a big smile on my face because it's like just what we thought we understood the brain. Turns out it's actually far more complex than we imagined.
I think it's also a great way to look at the data. I mean, I think I'm especially a fan of like how you could sort of like imagine that all of these neurons are kind of correlated with each other, that their activity actually drives another, and you kind of see this sort of patterns in space, and they're actually meaningful, that it's not just random patterns of activity, but something that's actually beneficial for behavior, and I think it's really cool.
Yeah, totally. Yes. So Jonas, you're going down the BCI route now. Neha, what comes next for you? What are you going to continue working on?
Hopefully a postdoc. That's next in line. Yeah, I mean, I'd love to continue working on this same sort of work that I do for my PhD, right? It's like I said, I really like the theoretical focus that we have in neuroscience, right? And I think it's really beneficial to look at we're at the stage where we're generating a lot of data, right? We have massive source of data now, and we have I mean, we're recording like if you look at like mice and non-human, like monkeys and so on, primates, right, and non-human primates, right? You see that there's a lot of data that's being recorded from different parts of the brain that we can look at, and we can kind of try and understand the behavior that we produce in terms of this, right? And obviously also try and relate it to something that's more meaningful, like I don't know, like motor behavior or like motor control and sort of stuff that's actually very important for like what's to come, right? Like robotics, BCI, prosthetics.
Hmm.
I think it's a field that's really evolving, and I'm happy to be a part of it, contribute to the science that's new and upcoming. Yeah, that's what's next, I guess. Yeah.
Cool. Yeah. It's definitely an ongoing journey. I like to think in terms of like, finite versus infinite games, and it seems like research is this infinite game, right? Like the more you discover, the more you realize how much there's left to discover.
Definitely. Yeah.
Yeah, I wanted to kind of maybe think about yeah, the journey so far. You're our first episode, both of you collectively, with the team, teammates together, I want to hear more about like the journey. What's good advice that you would give to researchers working in teams? Advice, maybe like what's bad advice that people should ignore? What was your experience? What kind of got you through it?
I don't know. I think it was pretty easy to work with Jonas, to be honest. I mean, we just ended up sitting in the same office, right? And um, yeah, and I think I've had no trouble, at least on my end, working with Jonas. I think it's very important to surround yourself with people who actually sort of challenge your views, and I think coming from a very technical view, I mean technical background, I had a different perspective on how to look at things, right? And I think Jonas constantly sort of challenged me in a very good way, right? Where he was always questioning about, okay, but what is this sort of data? I mean, for me, it's all about the data, and I look at the data and how cool the patterns are. And Jonas was always like, okay, but what does this mean neurophysiologically? What is the meaning of this? What is this sort of pattern doing that's actually important for us? And at the
Mm,
end of the day, I think it's like if he hadn't asked those questions, I would have never ended up doing these cool analysis to prove him, sort of like this is what I'm looking at. And I think that worked out really well for us. So
Yeah,
yeah.
I would agree. For me, the main takeaway of this and also other projects from the lab actually is talk to each other. Like usually
yeah.
these labs are organized in a way that basically everybody has their own little project going on. But from my experience, like also the way I work and the pace of my work increases dramatically as soon as I start talking about these ideas and actually verbalizing my thoughts and not getting lost in any like small analyses or small little bugs. So as soon as you start actually talking and discussing these ideas, it all makes so much more sense. And in particular, these 3 times in Tübingen were extremely productive. And I think the main part or main reason for this has like really been the lab culture and the way we discuss these
Yeah.
things.
I think we should really make use of the people around us who are also kind of looking at the same questions in a different way, right?
Yeah.
And yeah, this is great.
Yeah, thanks for sharing. No, that's been my experience as well. And part of the reason why I built this podcast is I want to increase the crosstalk between scientists in, similar or dissimilar domains, because I think it's at those intersections in conversation where interesting ideas come up. And so, yeah, we're coming to the end here. And so I kind of wanted to hear from you both in your respective research areas, like who do you want to talk to? Who do you hope reaches out? Who would be helpful to reach out to you? And this could just be based on your own curiosities or maybe like something a little bit more practical given the work you're doing now. What are your thoughts?
I mean, I enjoy talking to all kinds of people from very different fields, and also like experiences like this are extremely like valid or really important because you always have to try to get a bird's eye view on your own work and find a different perspective and not getting lost in any like technical issues. I mean, for me specifically now with the BCI work, I'm very interested in learning more about like of course like the whole machine learning behind it and stuff like that. And I really admire people who manage to translate this stuff to sort of more medically oriented neuroscientists like myself. And this is, I think, will be an extremely relevant exchange in the future. Yeah.
Yeah. I mean, anyone who's interested, right? I feel like the ideas can come from anywhere. And I think sometimes we're just stuck in our own research. We don't see what's right in front of us. And I feel like someone who's not really looked at the things the way we look at it have like maybe sometimes even a better perspective or even a better idea of what might be the next step, So anyone who's interested in the kind of data that we do, or like, I mean, just reach out. Happy to talk science anytime, right? It's it's a fun thing to do, I guess. Yeah.
Yeah. Yeah. Agreed. Well, thank you so much for taking the time to talk with me today. It's been quite interesting, and I'm sure I'll continue to learn more in this space. Thanks again.
Thank you
Thank you,
for having us.
Andre. Thank you. Yeah. And it was great having the podcast with you too.
Yeah,
We're very happy you reached out.
me too. ModernVivo designs best-in-class literature review software to help preclinical scientists identify insights from millions of peer-reviewed papers to design their in vivo studies faster. The ModernVivo podcast was written and produced by Andrés Velarde and Ian Levine in the Capitol Hill neighborhood of Seattle, Washington. For more information about our guests, please refer to our show notes. And if you have an interesting story to tell, and interesting science to talk about, reach out to Andrés at modernvivo.com to learn how you can be a guest on our show. Thanks for listening. We'll see you next time.